Okay, we're live. Hi, everyone. Thank you for joining this Google Science, Google Plus Hangout on Air. I'm joined by Matthew Francis, who wrote an article on Mosaic Science this week about a really profound question: what is life? Hi, Matthew. How are you?
I'm good. How are you?
Good, thanks. And also, yeah, Matthew's lower third is not working because, yes, weird Google issues. So, Matthew, can you introduce yourself and tell us who you are, what you do before we jump in?
Well, I'm a science writer. I write mostly about physics and astronomy, but my background is physics. I actually taught college for a little while before moving into writing full time. Um, but I've always been interested in some of the broader questions about science in the big questions that often come up, and so I was thrilled to have this opportunity to write this article for Mosaic.
Awesome. So, how did you actually research this article, and where did you get the idea for writing it?
Well, the idea actually started last winter. I went to a conference for the American Association for the Advancement of Science, and one session was on roughly defined astrobiology, which is, it sounds like science fiction, but it ends up being a lot of people working in several different disciplines. There's a lot of people who work in the laboratory with biochemistry. There's people who work out in the field looking for extreme microbes living in environments that are hostile to organisms like us, and it involves people who actually are astronomers looking at other worlds. And so I heard at this conference it actually was mostly biochemists, and I started thinking, okay, well maybe this would be an interesting article. Let's talk about what is life sounds like a philosophical question. And obviously it does have a lot of philosophical aspects and some very interesting ones. But it also has a lot of scientific aspects. We do not have a rigorous scientific definition of what life is. And so I started thinking, okay, well, this would be an interesting article. And so I spoke with those biochemists, but I also thought, well, let's look for a place where I could go and see field biologists and uh, geochemists looking at these extreme organisms. And I found a research group working, a NASA research group working at a lake in Canada called Pavilion Lake. It's in western British Columbia, about 4 hours drive out of Vancouver, so it's quite remote, but not extremely remote like some of these sites are. That was the other thing, is I needed to be able to get to it without without having to go to Antarctica, which
Yeah.
is, but um, but there, I just talked with all the researchers. I got to spend several days watching the work that they do, and it was a very enjoyable time.
So, why is NASA interested in a Canadian lake? It doesn't seem to fit with the remit.
And they wouldn't ordinarily be interested in a lake like this, because if you look at it, if you look at just the lake, it's very beautiful, but it looks like any other mountain lake you might see in British Columbia. It's in this narrow valley. It's pretty blue water, but underneath the surface, there are all these structures that look very much like coral reefs. It's amazing. They look like blobs of coral. Some of them look like artichokes. um, it's amazing looking, and it's all over the bottom of this lake, down to the point where there's no more light. And uh, and those are not corals; those are made by bacteria. There are bacterial mats that cover these things, and the water is full. Actually has a very high mineral content. And what happens is, as the bacteria metabolize, they extract the minerals from the water and build these structures. Well, the thing is, these bacteria are related to some of the earliest organisms we know on Earth. These are cyanobacteria, which are sometimes known, if you're like me, you learned them in school as blue-green algae.
Yeah.
Um, but we know now they're not algae, because algae is a type of plant. These are bacteria. And what they do is they take in, they photosynthesize like plants do, but they're bacteria. So it's a very simple cell structure. But also, this is something cool that I didn't have space for in my article. They actually are several types that work together. This is a colonial organism. They cooperate chemically to live. And so this is why NASA's interested.
Right.
Because these are organisms that live in an environment that is not so extreme. It resembles, the organisms resemble organisms that existed very early in the history of life on Earth, and the environment is possibly similar to perhaps an ice-covered lake when Mars was wetter than it is today. It might resemble these ice-covered moons.
Yeah.
In very, very, very broad sense, these ice-covered moons in the outer solar system, like Europa or Enceladus. And so NASA's interested in it for that reason. So it's very interesting to look at this, because of course this environment is not like some of the places that that we see. Shark Bay in Australia is one that comes to mind. That's a very saline environment. It's very it's very salty. It has almost no, if memory serves, it has almost no oxygen in the water, and this is very different than Shark Bay. So that's another thing that makes people interested. Is like, oh, well, why is this different? Why do these communities exist here and not in the next lake over? And it's all these interesting open questions.
So the title of your article, "What is Life?" that's the same as the title of the book by Erwin Schrodinger, the famous cat in the box experiment guy. So what does he have to say about this question?
Well, Schrodinger was looking at this question in the 1940s, and he was thinking about it. He's a physicist, so he was thinking about like a physicist does. And so he was thinking, okay, what is it special about life that makes it different from other types of things? Physicists had been really interested in crystals. One of the big triumphs of quantum physics, which Schrodinger, of course, was deeply involved with, was understanding the structure of solid materials. But life is different somehow, because crystals are good at reproducing themselves. Anybody who's done made rock candy or made crystal over saturated water, dumped too much salt in to make the cloudy crystals in water, knows that it's easy for crystals to reproduce themselves. You don't have to do anything. They naturally link together and make these crystal lattices.
Yeah, it's the laws of the universe, how the atoms just join up.
And it's simple as can be.
Yeah.
And Schrodinger said, okay, well, life somehow, knowing what people had learned about genetics by that point, and also about evolution, life doesn't perfectly reproduce itself. There's little changes between generations. And he said, well, this means that life must involve aperiodic crystals. So instead of perfectly repeating patterns, there must be some sort of code where the pattern changes just a little bit.
Okay.
And so this was in the decade before DNA was discovered. And so he's considered to be, one of several scientists who predicted what DNA must be like before it was discovered. He thought it was probably a type of protein. We know now he was wrong.
Yeah.
But it was still an interesting concept.
Yeah, okay. I mean, even Watson and Crick, at first they thought it would be a protein, but it was later that they realized it's a nucleic acid. So what is the definition of life?
Well, so that we actually don't have one. We have some basic ideas. Mm-hmm. It's not as though we're completely in the dark about it, but we don't have a single scientific definition of what life is. We know evolution is part of it, mm-hmm. because again, crystals can reproduce, but they don't change, they don't adapt.
Yeah.
So adaptation is obviously part of it. Metabolism is obviously part of it. It's not just a matter of reproduction. It's a matter of, or self-replication, because again, there's a bunch of crystals that do that. Life also takes in chemicals and
changes them.
Yeah, changes them. It exhales. Life breathes in and out. It poops. There's all sorts of things. It's obviously on a microbial level that doesn't quite make sense. But if you think about a yeast, okay, think about beer, or even yogurt. What you have is these microorganisms eat sugar, and and the waste product that they produce, the byproduct, changes the chemistry of their environment. It produces carbon dioxide, it produces alcohol, it produces the acids that make yogurt what it is. Okay, so we know how that works. We can see this on the microbial level. I mean, it's obvious for us too. On the level of humans, metabolism is very important. If you breathe into a plastic bag, you start running out of air very rapidly because you use up the oxygen. Not recommended, obviously, but it's the reason why you have to be very careful, and why space travel is tricky, and why traveling to the bottom of the ocean is tricky, is because we need we change our chemical environment rapidly enough that we can actually suffocate ourselves just by breathing normally if we use up the available source of oxygen.
So with that definition, crystals wouldn't count as life because they don't really change the environment.
Right.
But
and DNA by itself isn't life.
Yeah. So where does it get fuzzy? Because obviously, with a human with life like us, we can come up, wave hello, yes, I'm alive. But obviously, say with viruses, does it get fuzzy? Is a virus considered life because a virus can't exist outside a host cell?
Yeah, and that's exactly where it gets fuzzy. Because viruses have DNA or RNA. They do reproduce. They require a cell to reproduce, but they do reproduce, and they do evolve. They undergo natural selection. They all of Darwin's stuff.
Sometimes too fast for us to keep up with. That's why you need a new flu shot every year. So
exactly. And that's exactly why they are fuzzy, because they don't reproduce on their own. They are not alive in
the traditional sense.
But neither are they non-living.
It's that gray area between life and death almost. Because a virus out on a shelf will die, and that would be the end of it. I think it was the polio virus, I'm not sure, but it can't exist at all outside a host. So once you get rid of any natural infections of polio, that's it, it's gone.
Which is lucky for us in the case of polio. Of course, other viruses are a little more stable. But
what about prion proteins? Because that's something I've always been fascinated by, and that seems even fuzzier than viruses.
Yeah, and that's actually a topic that David Lin, one of the biochemists I spoke to, talked about. But prions reproduce also. They require a host. They're proteins. They reproduce, say, in the most infamously inside brain tissue, and that's where they cause things like mad cow disease and the human version, which is Creutzfeldt-Jakob disease. I got it that time. I totally forgot that one, blanked on that name the other day in an interview, and it's like, what in the world is it called? But those are conditions that are caused by these proteins that reproduce themselves. They do exist in a beneficial environment in certain yeasts. So they aren't automatically harmful, and actually viruses aren't automatically harmful either. Mammals use virus DNA to suppress immune response so that mothers don't reject their babies.
Yeah, and you get viruses that kill bacteria, which helps us. So
Right. Yeah. There's, in fact, that's an area that people have studied a great deal, is understanding how do viruses kill bacteria, and can we use those beneficially for us.
yeah,
But prions, I don't think anybody's found a beneficial use for humans yet, but the fact that yeasts use them beneficially says that these aren't automatically harmful things. They're just another way that we've got this kind of gray fuzzy area between life and non-life.
yeah,
And that raises an interesting question as well. We said that Schrodinger thought that genetic material was protein based.
yeah,
Some researchers are looking at the question, okay, well, right now, life uses a fairly complex process. DNA and RNA use each other to make proteins, and proteins enable the process of DNA and RNA to
yeah, because
have a safe
proteins are
environment.
enzymes, and you need those enzymes to make the DNA and RNA again and again. It's almost it's one of those chicken and egg questions. What came first?
Exactly. And so some people are saying, okay, well, how did that process get started? And one possibility is that maybe proteins originally reproduced themselves, kind of like prions do. But at some point, they learn to bootstrap in these nucleic acids, these genetic molecules. And so that's a possibility that people are investigating, that maybe proteins could reproduce themselves.
Yeah. One thing I've heard of, though, is the RNA world hypothesis, because
Right.
they think that RNA is older than DNA, and that RNA came first, and DNA is almost like a more sophisticated version of it. And you have DNA that's built you get DNA, and that makes an RNA template that makes the protein. So RNA has almost become an intermediate step. What do you think of that, and how does that fit in with the proteins came first idea?
Well, that may actually, I don't know if these are incompatible ideas,
Okay.
because I think the research is still very complicated and a lot of questions being asked. But the RNA world hypothesis is definitely one that is interesting because, again, like you were saying, RNA is simpler than DNA.
Yeah.
But RNA is still a fairly complex molecule. And so we're still left with the problem of where did that RNA come from? And that's another branch of research, is something called XNA, where you have an X instead of the R. The R stands for ribose, which is a type of sugar, and the D in DNA stands for deoxyribose, which is ribose without an oxygen atom.
It's both different types of sugar.
Yeah. But the thing is that those are still sugars, and so the more different than sucrose and things like that, but it's still chemically a sugar. And so some people are looking into the possibility, okay, could you build a genetic molecule using a different type of sugar? And that's what that XNA means, is that you have X could be other types of sugars. And could you have a sugar that's simpler, that looks like RNA, and could sort of have been the beginning of RNA? So in other words, RNA didn't come first. There was this simpler molecule that came before RNA, but RNA was more successful, and DNA arose from RNA, and then everything else followed.
So it's almost survival of the fittest at that molecular level. So there were lots of different types, but maybe RNA is the only one that made it.
And that was a hypothesis I had not honestly even thought about until I started working on this article. But when you start thinking about evolution on a molecular level, when you start thinking about selection on a molecular level, you have to ask, why does life look the way it does?
Yeah.
We all every organism uses DNA that we know of. That is all based on proteins. It all uses the same chemical building blocks. Did it need to be that way? And the answer is maybe, maybe not.
Yeah.
And so
We
one of
don't
the
know if it's actually the best way, or maybe it was like an accident, like the comet that, sorry, the asteroid that wiped out the dinosaurs, and that's why we are here now.
precisely, and that's something that I raised in my article, is the notion that there may have been competition between different chemical modes of life when life first arose on Earth, and ours was the winner.
Yeah.
But it didn't need to necessarily be that way, and that was a point that it really struck me as being kind of poignant, was the notion, we think about extinction as being on the level of species. A species dies out, but if there were more than one, if there was more than one way of chemical life,
Yeah.
3.5 billion years ago when life arose on Earth, assuming that new result that came out last week doesn't hold up, but that pushes it back. But anyway, whenever it arose, then there was an extinction not just of a species, but an entire possible mode of life.
Yeah. That's one of those mind-blowing things when you think of it. You're like, oh yeah. But then it also sounds really simple, because why would evolution just operate at the species level?
Right.
Yeah. So moving on to something that you talk about in your article, about biosignatures. What are biosignatures, and why should we be interested in them?
Well, that gets back to the metabolism.
Okay.
If you were to, okay, so if I were to take the room I'm in and seal the windows and doors, okay, and sit in here for 5 minutes and then leave, okay, you would be able to take various chemical tests of the room that I was in. You'd be able to see, okay, the oxygen level is slightly lower, the carbon dioxide level is slightly higher. There are other chemical traces that I would leave in the room just because I was there. And you don't even have to do it on a sophisticated chemical level. You could do it with a dog. You could do it with, all sorts of things like that, because I leave chemical traces. That's all a biosignature is, ultimately, is those chemical traces that life leaves behind. If you have a bacterium that dies, it leaves chemical traces behind. Metabolism leaves chemical traces. There's all sorts of things like that. So the question is, once the organism is gone, can you go and see if it was there, if it was ever there? There's the classic example is stromatolites, which are a type of rock formation. Okay, those coral reef formations I mentioned at Pavilion Lake,
Yeah.
are called microbialites. And stromatolites are a type of microbialite that basically all it means is microbialite is a structure that was built by microbes. A stromatolite is a microbialite that has these layers. There's a picture in my article of these sandwiched layers.
Yeah.
The thing is, that could be a natural formation, or I should say not a natural formation, but a formation that wasn't produced by life. How do we know it was made by life? You look for those biosignatures. You look for the chemical traces of metabolism. You look for isotopes that, different types of the familiar chemicals, but life utilizes certain isotopes and not others. You look for those and see what the ratios are.
It's almost like looking for the footprints of life and saying, there's no life here right now, but there's a footprint here, so that must mean there was life at some point or the other.
Exactly.
Yeah.
And that's what makes it interesting, because if you find something that looks like a stromatolite or a microbialite, if you find something that looks like a Pavilion Lake artichoke on Mars,
Yeah.
okay, that would be exciting. First of all, because the structures look really cool.
Yeah.
But if the question is, did this come about through the action of water and time and windstorms and all the types of chemical processes that happen on Mars without microbes, or were there microbes involved? And the answer would be depend on whether we could find these biosignatures.
Yeah. It's
recognize them for what they are.
Yeah, exactly. It's almost like if you find a cliff somewhere and it's shaped a certain way, is that because of the waves pounding against it, or was there like some kind of animal that was scrabbling there?
Right.
Horrible example, but it's like, is that an animal doing it, or is it millennia of an ocean doing it?
Yeah, or there's a fine example is, if you're walking in a field and you come across a rock that looks like an egg. Okay,
Yeah.
is it a fossil? Is it a fossil egg, or is it a stone that just happens to look like an egg? There
Yeah.
are ways to test that, and it's the same. There are many examples of that. And how do we tell the difference between a rock that was shaped by life and a rock that was shaped by chemical processes that aren't living?
Yeah.
And that's tricky.
Yeah, and can you ever get a definitive, yes, this is 100% answer, or is it always, oh, the jury is out?
Well, on Earth we can.
Okay.
Because we do have at least in many cases.
Yeah.
I mean, there are cases where it is difficult, and some of the biogeochemist, which is a great portmanteau word, the ones I talk to mention that even if you know that there are microbes present in some systems, it isn't always easy to tell which chemical processes were produced by life and which weren't.
Yeah.
Just because there's complexity in the system, it's hard to do.
Yeah. So one thing that I was really struck by your article is you talk about, well, it's this concept of redundancy, and I wonder, do you think that redundancy is something that's essential to life? So you talk about how carbon, for example, makes lots more molecules that are used by life, and we know with DNA and RNA, there's way more nucleotides than there are actually used in it. We know the genetic code is redundant, so there's more coding than actually you can get through proteins. Do you think that almost provides fodder for evolution to happen because there's all these spare parts lying around, so to speak? Or do you think is it useful for life somewhere else? Could it be that life somewhere else looks like it's made up of all the spare parts that we're not using on Earth?
Well, that's a good question, because we do see, like you said, we do see other amino acids, which are the chemical building blocks of proteins.
Yeah.
We see different ones in space than are used by life on Earth. The question is, can you build proteins out of them, and could life use those elsewhere? And we don't entirely know. The fact that carbon is very good at making these polymers, these long chains of molecules that are good for proteins, that are the fats that build up our cells. They're without carbon, it would be very hard to make these polymers. It just doesn't work.
Yeah.
And so the question is, how much does that actually have to do with the success of life and evolution? And the answer seems to be probably does, but it's hard to tell until we could actually build, unless we could figure out, either build it ourselves or find it elsewhere in the solar system. Yeah. It's hard to tell exactly what could be used. Now, as for redundancy in the genetic code, I know that this is something that's way out of my field, but I know that I've read a great deal of speculation on this. Obviously, some of that is probably a certain extent that some of that is an accident of evolution. It's just that as organisms evolve, as DNA changes over time, it just builds up all of these extra letters,
yeah.
Which are they don't get lost, they just stick around.
Yeah.
But that can also make organisms robust in some cases, if my memory serves. And so
Yeah.
The answer to that seems to be yes. Redundancy is good.
Yeah, in the case of DNA, it's almost like a safety mechanism, if you will, because with DNA, there's 3 letters that code for each amino acid, and the 3rd letter can change.
Right.
But the amino acid can stay the same. That's the redundancy in the genetic code. So if you have a mutation, you should probably hope you get it in that every third letter, because that way nothing will change. The amino acids won't change, and the protein wouldn't change, and you wouldn't get cancer or be born with a arm growing out of your head, kind of thing. So I think we're almost up at the half an hour, because this was such a fascinating talk. Do you have anything to add that you couldn't, include in the article, or you just thought of? Because it's such a, it's such a simple question, but it feels like the answer is just so vast. You could spend, you could write a book about it.
Well, a book is something that is definitely on my mind. But one thing that I guess I would say is part of the reason I've talked so much about microbes and microorganisms and chemistry is because if we are looking for aliens, if we're looking for life on other worlds, we're probably not going to be looking for, the type of things you see in science fiction movies. You're not going to see Vulcans or green, green bug-eyed creatures. You're going to be seeing microbes of some sort. Whether those microbes look like the ones we see on Earth, we don't know yet. But they're almost certainly going to be microbes, because most life on Earth is microbial. There's a lot more bacterial species than there are vertebrate species. The little things are the ones that are the most successful and fill the most niches. And so when we're talking about looking for alien life, that's ultimately what our best bet is going to be, is trying to find these microbial mats under, or signs of these microbial mats we see in Pavilion Lake elsewhere. And so, but I don't think that's any less exciting in its way, because it does tell us so much about the nature of life. Finding life anywhere else, even if it is totally different than life on, or actually especially if it's totally different than life on Earth, it tells us so much about life itself. And that's why it's worth looking at.
Yeah. Awesome. Thank you so much for that fascinating explanation of why you wrote it. And I hope everyone watching has a chance to read that article, because it just makes you think, and that's something I love about writing like that. So thank you for joining us, and yeah, good evening, goodbye, wherever you are.
Thank you very much.
Matthew. Bye.